to revisit the structure of glycogen, right? Briefly, what does glycogen look like? It will have 1,4 linkages, right? 1,4 glycosidic bonds. Yes, and every 10 to 15 glucose units, there is a 1,6 bond. So there is a 1,6 bond like this. 1,6 glycosidic bond. So right, so this every 10 to 15 carbon units, there will be a 1,6 branch like this, and this branch will continue, right? So this is briefly glycogen structure. So then when blood glucose is depleted, glucose is the primary energy, and when primary energy that is needed for red blood cells, that is needed for brain, that is needed for muscle contraction. When the blood glucose is depleted, the glycogen stored in the muscle and liver is converted to glucose molecules. So if the glucose in the blood is depleted, then the glycogen stored in liver and muscle should be converted to glucose. This process is known as glycogenolysis. Right? So how does this work? The first one, so we have this huge chain, and every 10-15 carbons. So let me draw the chain differently here. So let's say there is a chain of, let's say, 12 carbons, and every 12 carbons you will have a branch. So these are all 1,4 linkages. This is a 1,6 linkage. So every 10 or 15 carbons there is a branch, right? Technically this is what, symbolically, this is what our glycogen is. So in the first reaction of glycogenolysis, glucose molecules are removed from the ends of the chain using to yield glucose 1-phosphate. So this, each of these 1,4 linkage is broken one by one. One by one, this 1,4 linkage is broken, and one glucose at a time is taken out, and it is phosphorylated to give glucose 1-phosphate. Right? Now, till the last molecule, till the last glucose, then another enzyme comes in. This is known as 1,6-glycosidase. 1,6-glycosidase, and I'll use another color. 1,6-glycosidase. What this does is it breaks in this 1,6 branch. It breaks in that 1,6 bond. And then once you break that 1,6 bond, each of these is again connected with 1,4 linkages. So in the same, just like the first reaction, each one of these is broken into glucose molecules, and they are phosphorylated. Once this is obtained, this phosphate group moves into, so there is a CH2OH here. This, in the rearrangement reaction, the mutation, the rearrangement reaction, the phosphate group migrates here. So when that phosphate group migrates, Well, that is again with the help of the enzyme phosphoglucomutase, and we have talked about that before. So once that happens, phosphate is now here, right? And if you remember, this is the glucose 1 phosphate, and this is a part of glycolysis. So this is one of the molecules in glycolysis. So what happens? This enters glycolysis, and then say this enters the glycolysis cycle and goes into the step 2 of glycolysis. Now, if the glycogen was present in liver, see, glucose can move across cell membranes, but phosphates cannot move across cell membranes. So the cells in liver and kidney, if they have glucose 6 phosphate, then glucose 6 phosphatase will hydrolyze glucose 6 phosphate to glucose. So what will that do? Instead of the molecules entering glycolysis, they can't enter glycolysis because they can't pass the membranes because they are in the liver. So CH2OP, this is your glucose 6 phosphatase, this is your glucose 6 phosphate. So glucose 6 phosphatase will convert this to glucose, hydrolyze it to glucose and phosphate, and this goes into the bloodstream. So also remember, I am not drawing the OHs and Hs in these molecules.